all engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
Discoveries.
Advances.
Questions. Research.
technology.
Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to The Naked Scientists podcast. This is the programme that brings you the biggest breakthroughs and also talks to the major movers and shakers in the worlds of science, technology and medicine. I'm Chris Smith. Today we're putting one of the world's most pernicious parasites under the microscope, malaria, and Titan of Science, Jane Carlton, is going to be our guide. Jane Carlton was born on the 7th of January 1967 in Scotland's capital, Edinburgh. She attended St George's School for Girls and then completed a degree and a PhD in genetics at the University of Edinburgh, graduating in 1995 before moving to the US to take up a research position at the University of Florida, where she used her skills in molecular biology to produce the world's first full genome sequences of the malaria parasite. Since then, she's worked at NIH in Maryland and at New York University. Jane was recently elected as a Fellow of the American Academy of Microbiology, and she was also awarded the American Society of Parasitologists Stoll Stunkard Memorial Award in 2010, made a Fellow of the American Association for the Advancement of Science in 2012, and named Honorary Fellow by the London School of Hygiene and Tropical Medicine in 2024. She's now Director of the Malaria Research Institute at Johns Hopkins University in Baltimore, Maryland, a post she took up in 2023. That is quite some CV, Jane. It's wonderful to speak to you today. The focus of your career has mostly been malaria, which is one of the world's highest impacting parasitic infections. So why don't we begin there? What actually are parasites? What's the definition?
Parasites are fascinating organisms. They live in or on a host, and usually to the detriment of that host. That can be through making the host unwell, or perhaps reducing the ability of the host to reproduce, or even changing the behavior of the host. And the parasite that I work on in particular, the malaria parasite, is a protozoan, a single-celled organism.
What took you down that path, though? Tell us a bit about the young Jane Carlton that got interested in parasites in the first place. How did that happen?
It wasn't initially parasites that I was interested in, actually, but genetics. And that's because my brother went to university and he came back with this new book all about genetics. And I really liked the front cover, which showed some chromosomes. And I thought, well, how cool is that the biology of an organism can be determined by these things called genes, and then how they're inherited can really affect their biology. So it was really through genetics and through my brother that I got first interested. And then when I went to the University of Edinburgh, there was quite a well-known and well-thought-of group that worked on the genetics of malaria parasites. And that's really when I got into the world of parasitology.
But how old were you when you picked up that genetics book then?
Actually, I think I was about 10 years, or maybe 8 years old. I can't remember. I still have it, actually.
You stole it.
I did. I've actually stolen a few things from my brother over the years, as he knows.
But you must be pretty unusual. There are not many 8 to 10-year-olds reading genetics books. I mean, that singles you out as a bit special.
Oh, well, I don't know. I mean, there's an interesting interpretation of the word special, isn't there? But no, I think my brother was the special one, bringing the book home, and that he actually taught me an awful lot about the world that we live in. So, no, I put it down to him, not me, really.
But obviously that must have inspired you, because presumably you then followed a science track through primary school, secondary school that would enable you to apply to Edinburgh University to do, because you did genetics there, didn't you? So how did you end up in that degree course? Was it just because of that book and then pursuing science at school?
Well, it was always an interest in the natural world around me, actually. So I was always a very much an outdoorsy girl. And I loved riding and going off by myself and investigating things. And so then it was really wanting to know more about the biology and the kind of ecology of many of the organisms that I came across and the plants in the real world. And then knowing how those developed and the role of genes and genetics. That's how I really got interested and decided to take that degree at the University of Edinburgh.
And it was that degree that then turned you on to malaria because you were applying that genetics interest to the malaria parasite. Because at this time, this must have been, what, the mid-90s? And although we take it for granted these days, I'll just sequence that and we read the genetic code of everything we come across, don't we? In those days, the Human Genome Project was still, 5 or 6 years away from reporting its initial draft. So this was very early on in the game.
Yes, it was. I was very fortunate at the University of Edinburgh in the Department of Genetics that there was a very large group, very well-known group of malaria researchers that used genetics to study the malaria parasite. And so that's really how I got involved and did my PhD doing genetic crosses between different parasite strains that had different susceptibilities to anti-malarial drugs. And I had to generate the first genetic map of a particular malaria parasite species because the genome had not been sequenced. And that was very painstaking and it wasn't that great a map. And that's really when I started to hear about news, especially in the United States, of an initiative to sequence the human genome. And I knew that there was a group at the University of Florida that was planning to sequence the malaria parasite genome. And so I jumped at the chance to try and join them.
And that's what took you to the States?
Yes, that's right. It took me to University of Florida. And I think many British people seem to have an attraction to Florida because of the wonderful weather. I went there both for the weather, but also because of this great group who were tagging the genes of certain species of malaria parasite.
There's more than one type of malaria, isn't there, that hits humans?
Yes, there are about 6 different species of human malaria parasite. The most prevalent, Plasmodium falciparum, which infects primarily children under the age of 5 in sub-Saharan African countries, is the most deadly as well and causes about 280 million cases each year and about 600,000 deaths. But there are other species as well. Plasmodium vivax, for example, is found in South America, but also Southeast Asia. And the really fascinating thing about that parasite species is it has a dormant liver stage. And so people can be walking around feeling perfectly healthy with this dormant stage, and that can then pop out years, months after an initial infection and cause the disease. So yes, there are actually about 200 different species of malaria parasite.
Which other animals get it then?
Lots of mammals, so non-human primates and monkeys. Also reptiles, lizard malaria is a huge issue too. And then also birds. So certain species of malaria parasite have been decimating those beautiful populations of colorful birds that you find in Hawaii.
Given that it's in so many different species and even reptiles, then, does that mean that in evolutionary terms, this is a really ancient bug?
It is. It's an ancient parasite. It was first described or known to have been described by Hippocrates, for example, who was they call him the father of modern medicine. And he described the symptoms of malaria and the fact that people tended to come down with it if they were close to marshes. So it's been known for a long time. There have been some very notable deaths due to malaria. So Alexander the Great died of malaria. George Washington had many episodes. Lord Byron. More recently, George Clooney actually had malaria too, I think when he was traveling in Ethiopia. And it was great because he then used his illness to highlight the severity of the disease.
What's the actual process of malaria? I mean, most people know it's connected to mosquitoes and it's something to do with blood, but talk us through the actual process of how malaria does what it does.
Well, it's a really intricate life cycle. So malaria parasites are transmitted by female mosquitoes of a certain species called Anopheles. And that's because it's female mosquitoes that take blood meals. They need blood in order to produce eggs. So malaria parasites get transmitted from the bite of a female mosquito in the saliva into the skin. And then those parasites migrate to the liver. And in the liver, they undergo cycles of reproduction. And then they burst out of liver cells into the blood. And it's actually those stages in the blood that really cause the symptoms and the diseases, fever and the muscle aches, the headaches, the chills that really lead on then to more serious conditions as well. And then in the blood stage as well, the parasite can form these sexual stages, like gametes, male and female gametes. And so when an uninfected female mosquito takes a bite, those stages will then move back up into the uninfected mosquito and they multiply, and then the parasite moves to the salivary glands. And so the whole life cycle is complete. So incredibly complicated if you can think about it, because all of those different stages we have to study. We want to know what genes are turned on and off. And that can be quite challenging because you can't necessarily grow the parasite for all of the different species. It's a very complex life cycle.
It certainly is. And to think that one parasite can get into all those different cell types, that insect cells, liver cells, blood cells, grow in there and also evade the immune system at all these different stages, both in the insect and in the person. It really is quite extraordinary, isn't it? Can you actually see it, though? When we look down a microscope, can you see malaria inside these cells?
Yes, you can. You do need a microscope, but you can see it in the red blood cells, for example. It's actually quite beautiful. I know I shouldn't say that for a parasite. So the ring stage of a malaria parasite looks just like a diamond ring, if you can imagine, with the big diamond and then the sort of platinum band as well. So you can see that quite visibly in red blood cells. And then you can see it in other stages too. So there's sort of a sausage crescent-shaped stage, which is one of the sexual stages before it's taken up by the mosquito. And then actually in the mosquito itself, there are certain stages called the sporozoid. They're very interesting too because they're very motile. So they sort of glide along a bit like a snake.
So how does it actually make people ill? When a person's got this thing, obviously they've got something growing in their cells, which can't be good. But how does it actually translate into illness, all of this?
Well, it actually doesn't translate into illness for several weeks. And that's, for all of your listeners, if this is the one take-home message, if any of them go on holiday somewhere and then come back, if they're sick 2 to 3 weeks afterwards, perhaps when they've forgotten about their holidays, to remember that they went to a malarious country. Because when they go to their doctor, to tell the doctor, I've been to a malarious country, and that will immediately trigger in the doctor an alarm that potentially this person could have malaria. And the reason for that is that the malaria parasite takes a good 2 weeks or so to develop in the liver and then in the blood before a person starts feeling sick. And then that sickness tends to be a bit like flu-like symptoms, but it's very important that people, seek the doctor's advice and get treatment because it can spiral out of control pretty quickly. And in about 1% of cases, it can go on to convulsions and major organ failure, and then that can lead to coma and to death.
Is that because it's killing cells, or is it irritating the body in other ways?
It's a combination of all of those, actually. So, in fact, the immune response against malaria parasites can form a very big inflammatory response, which sometimes is not helpful. But also, cerebral malaria is when the parasite travels to the brain and causes blockages in the brain and brain swelling too. And that's really what can cause the death.
I'll come on in just a second to how we treat someone in that position, how the drugs work. But you mentioned that with Plasmodium vivax, that can stay in the body for long periods of time. But once a person gets better, does that mean the parasite is gone? And are they immune after that? So it's a bit like if I caught measles, I would be immune for life for measles. Do I then have immunity against malaria? No need to worry in future.
Yeah, very good question. So for children living in African countries, once they get malaria several times, they will be immune. But as soon as they move away, their immunity can drop. So people who are not in endemic countries won't have natural immunity to malaria. They won't have that built up unless they're going back to those countries, many times. So immunity is not very well understood at all.
This is the Naked Scientist podcast with me, Chris Smith, and I'm in conversation with Jane Carlton, who's director of the Malaria Research Institute at Johns Hopkins University. Coming on then to how people actually manage it, what do the drugs do?
Yes, malaria is a treatable disease. I say this so many times. The problem is getting the treatment to people in time. We do have a range of drugs, artemisinin-based combination therapies or ACTs. Quinine is another one too. Unfortunately, the malaria parasite has become resistant to almost all drugs that have been developed. And so we do have to use the drugs in combination because the probability of getting a parasite that is resistant to two drugs is much less. But timing really is key. It's getting the treatment, it's getting the drugs, to those children, to those travelers in time.
Presumably, the genetics work that you were doing when you first made your foray to Florida and generated those genomes, that is going to inform how we understand how something works, because that gives us the genetic insight into the mechanisms of the parasite's operation, and that must open doors to looking for new drugs and so on.
Yes, indeed. Well, I do want to say up front, I led the projects to decode the genomes of a couple of species of malaria parasite, in particular Plasmodium vivax, and a rodent model. And then I was part of the initiative to sequence the genome of Plasmodium falciparum. But I have to say, it was a real team effort. I stood on the shoulders of giants. I know that sounds a cliche, but the whole group of us coming together to pool our expertise to get all of this done. And when we did, the result was fantastic, and I think has really blown open the field of malaria research. And the reasons for that is that we can really start to classify and identify all of those genes which are involved in important processes in the malaria parasite. So we can identify groups of genes which have expanded perhaps because they interact with the human immune system. We can identify genes which potentially have mutations in them that cause drug resistance. We can generate genetic markers now to be able to do population studies. We can see how certain strains are moving through populations. And then, of course, we can do this whole sort of looking for chinks in the armor of the parasite. So finding genes that codes for a protein, perhaps in a particular metabolic pathway that doesn't exist in the human host. And right there, immediately, you have a possible drug target because you have a protein that's produced by the parasite and not produced by the host. And so you can potentially generate an antimalarial drug against that particular protein, and vaccines as well.
I was going to ask you about vaccines. Where are we with that?
Yes, we are really at the beginning, actually. It's been incredibly difficult to generate a good vaccine for malaria. We have two new World Health Organization approved vaccines now, RTSS or Mosquirix as it's called, and then R21. And they're only just beginning to get into the arms of children in, I think, 25 countries. The one that I'm most involved with is through our project in Zambia. And the Zambian government only just a couple of months ago started the first inoculations in children there. They're using the R21 vaccine that requires 4 shots. So it does take some time, this whole course of immunization. And then at that stage, with so many countries now vaccinating, we'll really be able to get a grip on how good these vaccines are. About 3 or 4 years ago, when RTSS first came out, there were initial studies in 3 countries. The effectiveness actually was not that great. It was only about 30 or 35%. There was a reduction in the number of deaths and the number of hospitalizations too, and the number of children, getting malaria, as I said, but not as great as we would have hoped. Now, with this larger number of countries, we can really get to grips to see how well the vaccination is going to be.
Do we know why it's such a headache to make a vaccine that's working?
We do, yes. Unfortunately, it's because the malaria parasite has a couple of really crafty mechanisms to evade the human immune response. One of them is called antigenic variation, and this is where the proteins on the surface of the parasite change so quickly that they can evade any immune response, any antibodies that the host has generated against them. And then there's antigenic diversity. So many of the antigens, the proteins found on the surface, are very variable. And so the parasite is able to evolve very quickly against whatever we throw at it, actually. And that's why it has become resistant, as I mentioned, to almost every single anti-malarial drug that we've used against it.
Do you think this is a surmountable problem?
That's a very good question. I'm not entirely sure it is, actually. I think instead of just believing in one magic bullet, there's a combination of all of these that's required. So we've really been talking about controlling the parasite in humans, but also, of course, we need to talk about controlling the mosquito populations.
Indeed, because I was intrigued when I interviewed a young woman who'd just finished some research recently, and it made me think, because her approach was, we don't just want to treat people because mosquitoes are infected too. So rather than trying to kill the mosquitoes, which we know that's a pain and difficult, why don't we try and treat them for their malaria? So she was inventing chemicals that the mosquitoes pick up when they try to bite us from bed nets.
Absolutely, yes. And I think I know who you're talking about, actually. That research is key. I think that's very important. There are other approaches as well, such as engineering mosquitoes that malaria parasites can't infect. So you're not killing the mosquito populations, but you're preventing the malaria parasite from being able to develop in the mosquito. Also killing the malaria parasite in the mosquito, perhaps through producing a toxin. In addition, being able to develop better baits that can attract mosquitoes away from people, or better, mosquito sprays that will repel mosquitoes more can be developed.
You're now directing the Malaria Research Institute at Johns Hopkins. So how did that come about, and how do you balance the managerial/director side of that role with the fact you're a hardcore scientist at the same time?
Actually, I had the tremendous opportunity and was asked to apply for the directorship of the Malaria Institute just about 3 years ago. And I had known about the Malaria Institute for many years. It was first formed through a very generous gift from Michael Bloomberg in 2001. Many of my colleagues and friends ended up at the Institute. And so I really jumped at the chance when I was asked to apply, and joined the faculty 3 years ago now. So I'm still quite a newbie, I have to say, and loving it. We're a group of scientists in a state-of-the-art facility that we have in Baltimore, in Maryland, at Johns Hopkins, undertaking quite high-impact research on all facets of malaria: the mosquito, the parasite, the human host. Ultimately, the goal is to innovate new strategies that prevent and cure malaria. So it's a dream come true for me. It really is. So I don't find it too troublesome, I have to say, in terms of, the managerial and the leadership side. A lot of the time it runs by itself. And I think that's when you have a great collegial group of faculty who all work together towards a common goal. That makes it so much easier.
Shh, they'll give you more to do.
Well, bring it on.
But more seriously, I mean, the role of a director is not just obviously to run an institute, but to have a world vision, especially with a centre as prominent as the one you're running. So where are you pointing your guns at? What are your targets? What's in your crosshairs for the next 5 years? Where do you think the opportunities are, and what are you going after?
5 years, I have to say, is quite a short period of time, but I have drawn up a vision and a kind of strategic plan for the Institute. And we have about 16 or so big projects that we're starting to roll out, which are very exciting. A lot of those involve our field sites. So we work in several African countries, Zambia being one of them, but also Uganda, Kenya, Tanzania, and more recently, my own work has taken me to Ethiopia. We're looking to see what improvements can you make to places that people live in that would reduce them getting bitten at night. We are closely monitoring and rolling out a study to look at the implementation of one of the vaccines in Zambia, to follow a cohort of children as they get the immunizations and to be able to determine how well they're responding to the vaccine and what the immune system, how it's responding as well. And the understanding that the vaccines are not perfect, we're rolling out more studies on generating new vaccines, in particular using mRNA technology, and also trying to develop or identify new targets for vaccines as well. My own research in Ethiopia, we're trying to look at refugee camps and to see how the parasite spreads through those, in particular anti-malarial resistance is a big issue for one group of drugs in Ethiopia. And we're also hoping to look at the invasive Anopheles stevensii mosquito in parts of Ethiopia as well, which is really upending the epidemiology of malaria in the country.
Am I being over-optimistic by saying it would be wonderful if in the future we got to a stage where no one has malaria? Is it possible to get rid of this thing forever, or is that not feasible?
Well, I think that's the million-dollar question, isn't it? I'm hopeful, I'm always hopeful that we'll be able to eliminate malaria, if not eradicate it, in many of those endemic countries.
And just for clarity, how do those two terms differ? What's the difference? What's the distinction between them?
So eradication is completely getting rid of that particular disease globally, and a good example is smallpox. Elimination, on the other hand, is when certain countries have managed to rid the disease, but their neighboring countries, for example, may still have that disease. I think elimination is really very much in reach, and we know that 48 countries have been certified as malaria-free by the World Health Organization, and I can only see that number increasing. In fact, the world malaria map has shrunk significantly over the past 50 years. The issue, of course, is in countries where there is internal strife, perhaps internal wars or environmental catastrophes such as the flooding that we saw in Pakistan a couple of years ago. In those sorts of situations, malaria can easily rebound. And of course, of concern as well has been the reduction in certain US funding initiatives too, in particular the President's Malaria Initiative. So we're a little unsure in the next few years what we're going to be seeing, but I am very hopeful through the rollout of these new vaccines and vaccination programs that progress towards global elimination will be possible.
That book that you stole off your brother has changed the world a bit, hasn't it?
Well, I think genetics is just a fascinating subject and area, so it certainly changed my life.
The formidable Jane Carlton with the past, present and future of malaria. And we'll be returning regularly to talk about malaria on the programme in the future because we are teaming up with Jane and her colleagues at the Johns Hopkins Malaria Research Institute to bring you the Malaria Minute, in which we'll be offering insights from around the world into the important work that's going on to combat the disease and its impacts. So stay tuned for that in the weeks ahead. Meanwhile, do join us as usual on Friday when we'll be back with our usual roundup of the top science stories from across the world. Do please follow us on LinkedIn and Instagram for up-to-the-minute information on what we're up to, and we very much welcome your reviews and comments on Apple, Spotify or wherever you get your podcasts. My email address, on the other hand, if you'd like to write to me, is chris@thenakedscientist.com. Lastly, Huge thanks to all of you who are continuing to support us here at The Naked Scientists on a regular basis. If you would like to make a contribution to keep the show on the road, we would appreciate that very much. It's an enormous help. Please head over to nakedscientist.com/donate. I'm Chris Smith. Thank you for listening, and until next time, goodbye.